And we know all that. Thanks very much. And they also have importance, relevance for reasons that we can't yet imagine, right? So when the colleagues from the researchers who were working on hantavirus back in the early 90s and even before then, who were doing field biology in the mid to late 20th century, they had no idea, of course, in the 70s when they collected these Peromyscus specimens, that 20 years later, they were going to help identify a reservoir for a very dangerous pathogen. Okay, so that's not controversial, I don't think. What is controversial to a greater extent, I'm getting rid of the little sign on my thing. Okay, is this notion that that the natural history collections and biological collections that we have in general are the result of what I referred to in my title slide as, bad actors, right? And I want to read to you a couple quotes. This one, the first quote is from this article from the Humboldt Foundation magazine called Cosmos. I'm sure you've heard of the Humboldt Foundation, a really important supporter of science based in Germany. And well, you can see for yourself, the cover of this magazine issue from 2024 says, "Everything just looted, how colonialism is still affecting science and where supposedly self-evident facts fall apart." Let's see now. I can't read the top of the slide because of the screen share, but in any event, I'll just read the last part of this quote here from 2024. The aim of these collections, people from 18th, 19th century Germany, for example, or Britain, going out to corners of the world to collect stuff. Alexander von Humboldt is an example of that. The aim of these collections and research was to underpin the supposed superiority of Western societies. Another quote from a paper in the Journal of Natural Science Collections in 2018. I'll just read the last bit. "Museums were put in place to legitimize a racist ideology. By ignoring this history, they are continuing to do so." Now, I personally don't think that either of these quotes are accurate. I think they're more misleading than true. Obviously, colonial histories of rich nations over the past few centuries has had an awful impact in many regards. But Alexander von Humboldt is a pretty bad choice, in the case of the Humboldt Foundation, to call a representation of racist natural history collectors, in the sense that he wasn't in the majority in the 18th century, but he was one of the most consistent abolitionists throughout his professional career well into the 19th century. In any event, this is the kind of bad actor to which I referred in my title slide. So now I want to shift a little bit and tell you about some specific legal manifestations that have, in my opinion, made the collection of natural history items more difficult in recent decades. And the first one I'm going to point to is NAGPRA, and many of you will know about this, the Native American Graves Protection and Repatriation Act. This is just a quote from the National Park Service website. Since 1990, federal law has provided for the protection and return of Native American human remains, funerary objects, sacred objects, and objects of cultural patrimony. I agree with that, right? That's an important step that we have to be cognizant of the history of our collections, and just as a phrase, as a statement, I think that's entirely reasonable. Now, a couple examples of specimens that have been repatriated and indeed reburied include specimens like this human skeleton discovered in the mid-90s from the banks of the Columbia River in southern Washington State. It's Kennewick Man. Many of you have heard of this. It's 8,500 years old. This has been reburied, right? It used to be in the Burke Museum in Washington State. It is no longer. Anzik is the name of a family who owned some land in Montana. There were human remains there too, a little bit older, 12, almost 13,000 years old. Those too have been reburied. Now, we're not, at least I don't have time to debate the merits of these particular cases, but what I do want to do is quote a paper by Weiss in 2024 in the Journal of Controversial Ideas. We'll come back to that in a moment, but what Weiss says, and I quote, is the repatriation process has caused the cessation of human remains research at California's public universities. So when I came across this quote fairly recently, I thought to myself, well, this is just a a disgruntled former employee of of a California state school being angry, and I don't have to take it seriously. But I did follow up a little bit, and I got in touch with some colleagues who work museum and university collections professionals in North America, and I asked them about this Weiss article. And I was told, I'm not going to share the identity of these people. You'll just have to trust me for a moment that this is, real museum professionals who care about collections, who are writing this stuff, that Weiss 2024 was not an exaggeration. I'm going to quote briefly from one communication I had with a colleague in North America, and I quote, "Removal of all human remains from teaching spaces and collections has happened. It doesn't matter where they come from. The bones get put away until our campus NAGPRA office can identify a descendant group that wishes to receive them, or they decide to do with unclaimed remains." And this is not just human remains. The quote continues, "This applies not only to the human bones, but also to anything else associated with them. Fossils, plants, animals, and other natural history objects may also fall under cultural patrimony. Even in the best of circumstances, it is another layering of permissions and paperwork that researchers must submit." Now, I had nothing to do with the Museum of Southwest Biology collection of Peromyscus from the Four Corners region back in the 70s, 80s, and 90s. My assumption in that case is that there was a perfectly good collaborative relationship that those scientists had with the Navajo Nation, for example, the different tribal groups in the Four Corners region. For all I know, there were tribal members of that scientific team who were themselves scientists working on this project. I have no doubt that this was a perfectly harmonious relationship. The fact remains, though, any application of NAGPRA in which it's slightly harder to collect stuff means that there's going to be less stuff collected, right? And so imagine, if you will, for a moment, that those, back in the 70s, those researchers had to contend with NAGPRA. Would they have collected these specimens of Peromyscus? Maybe, maybe not. It would have been slightly harder. And, who knows today what we're not collecting that in 2 decades' time would have been really crucial to identify a reservoir for some horrible pathogen. I'll just leave you with that thought for a moment, and I'm going to march on to try and finish in time maybe for a question or two and tell you about the Convention of Biological Diversity. Now, this has a heart of gold, right? This has really noble intent, but the application of the CBD and its sort of somewhat later version, the Nagoya Protocol, to try and enforce it more strictly, has had some really negative consequences. And I have to say, there's 1 or 2 sessions that I've heard in this meeting, which I think people are just being naive, and you're not taking into serious account what these guys said back in 2018, Prathapan et al. And I guess I'll just read to you a little bit from this Prathapan et al. 2018 letter to Science. Since the convention came into effect, and especially after the Nagoya Protocol led nation states to step up legislative processes to tighten their control over genetic resources, obtaining permits for access to specimens for non-commercial research has become increasingly difficult, right? Now, why does that happen? Well, one interpretation, which is what I think, is that the politicians and the lawyers and all these governments and all over the world are thinking that when a biologist shows up somewhere in the Amazon and collects some specimens, that they're a few weeks away from patenting some gazillion-dollar drug, right? And they want to cut. The dollar signs show up in their eyeballs, right? And who can blame them if that were the case? Of course, that's not the case. Basic science is separated by decades from any kind of patentable, profitable thing that a pharmaceutical company might develop. And this is sort of reflected, I think, in a very good article by Ronai and Griffiths in 2019, Trends in Molecular Medicine, where they point out that in terms of basic science, it's nearly impossible to precisely quantify the benefit of a specific basic research product, right? So there's a real disjunct here that I think is worth pointing out. I also want to point out what several of our colleagues in Brazil have been saying for some time. This is an example of that 2018 paper in the Annals of the Academy of Sciences in Brazil, signed. This is over 60 different university collection and museum scientists based in Brazil. And I'll just read to you the first sentence of that paper. Quote, "Beginning in November in 2018, Brazilian legislation regulating access to genetic heritage and associated traditional knowledge will cause a bureaucratic collapse of biodiversity research in Brazil." That's the first sentence. This is 2018 is a long time ago. Is this still the case? Well, I'm in regular contact with many of my dear colleagues, working professionals, scientists in Brazil. This is an extract from an email I got. Quote, "We continue to have the same issues, excruciating bureaucracy without the benefit sharing part of the Nagoya agreements." That was in an email to me, a few months ago. So finally, I want to share with you an example from our botanical colleagues, our botanist colleagues. This is a paper from Kano et al. This is a group based at my university, Botanical Gardens at the University of Cambridge. And what you're looking at here in their paper, Insights from a Century of Data Revealing Global Trends, it's basically a metadata where they've taken accession data from botanical gardens. Now, these are slightly different from natural history museums in the sense that they have to keep their plants alive, of course, right? So it's not like, our little zoology museum here at my university. You find a dead rodent, you stuff it, put it in alcohol, whatever, and that's it. These guys have a regular throughput of materials because all their collections are alive. And what you're looking at here on the y-axis is the number of new accessions. On the x-axis is the year. And in this case, you're looking at wild accessions, new accessions into botanical gardens. And keep in mind, of course, that the majority of these major botanical gardens are in the relatively non-diverse countries, places in the United States, in Europe, places that aren't nearly as rich in terms of biodiversity as for Brazil, for example. There are botanical gardens that are in the global south that are contributing to this. Nonetheless, you can see when the Convention of Biological Diversity came into effect in the early 90s, the number of wild specimens that are going into these botanical gardens is dropping like a stone. And here is a similar kind of phenomenon. Non-native is here in blue. The accessions drop with the enacting of the CBD. There's this odd little blip here, but of course the overall trend is the same. The rate of acquisitions is decreasing post CBD, and Nagoya 2014 is right here. That rate is becoming exaggerated post Nagoya. And of course the conclusion of Kano et al. In 2025, and I quote, "The inclusion of globally threatened plants in living botanical collections has been negatively affected by biodiversity legislation." So that is the end. I'm going to leave my conclusion slide up, hopefully for a moment or two, so you guys can see it. I apologize for being such an alarmist, but I really wish more of my colleagues, including you guys listening, would worry about this. I'm really worried. Okay, I'm finished. Thanks.
Thank you so much. Thank you for your understanding as we have limited time. Thank you for your wonderful presentation. But right now, our presenter is Zoe.
Shall I stop sharing?
Yes, sure. So you can start your presentation.
Thank you. I'm sharing my screen now. Can everyone see my screen?
Yes.
So thank you everyone for coming today. I'm really excited to get a chance to talk about my research on the evolution of thermal performance curves under empirical climates. And I worked on this with Dr. Ji-Sun Min and Dr. Katie Lauderhouse at Northeastern. So to open up the broader issue, Drosophila zuckii are an invasive fruit fly that are native to Southeast Asia, but have recently expanded into the United States and expanded their global range limits. And they're, unlike other species of Drosophila, they're classified as invasive because of their propensity to target ripe fruit, which leads to devastating impacts for the agricultural industry. And one of the perplexing questions about their range expansion is their ability to establish themselves in the Northeast United States, which has extremely cold winters, unlike their original habitat. So this leads to the question of their potential for adaptation in changing their thermal limits. So one of the tools that we can use to address this question, especially in the context of anthropogenic climate change, as mean temperatures, variances, and thermal extremes are changing is thermal performance curves. And thermal performance curves are model performance as a function of body temperature, which for ectotherms we can assume to be ambient, equal to the ambient air temperature. And so thermal performance curves capture the range of non-lethal temperatures that an organism can experience, with B or breath denoting that range, and then CT-min denotes the critical thermal minimum, and CT-max is the critical thermal maximum. Beyond these two limits, fitness approaches zero, and the thermal optimum is denoted by T-opt, which is we measured as 2/3 of the breadth because of the thermodynamic benefits of having a warmer body temperature. So one of the ways that we have seen the adaptive potential manifest in these invasive species is through seasonal adaptive tracking. And foundational studies have found this in Drosophila melanogaster and other Drosophila species. And you can see that stress tolerance and fitness-related traits, like reproductive output, experience directional selection over the season. So their trait limits will be different from the summer through the fall. And we can also see allele frequency shifts as the spring and fall, or how it oscillates throughout the spring and fall seasons. And typical theoretical or foundational theoretical work on the issue of seasonal adaptive tracking has tried to explain the balancing polymorphism that occurs. And some of them invoke theories such as like seasonal dominance switching, but these can be somewhat restrictive because they model the traits directly affecting fitness instead of letting the traits evolve to fitness itself. So we came up with a model that takes the typical thermal performance curve framework, but then we're imposing these physiological constraints on the CT-min, breadth, and CT-max. So any extreme trait value of these will impose a serious physiological penalty. And we use a multiplicative thickness, and from there we can get the overall fitness of the thermal performance curve. And so we extended the analytical framework into the quantitative genomics framework. And so we incorporated this in SLIM, and we had across the genome 2 neutral regions flanking a region in which beneficial mutations were allowed to occur. And we had, for every mutation, at a mutation rate of 10 to the -7, a 50% chance of either affecting the CT-min trait or the B trait. And if you remember from the original framework of the TPC, we can calculate both CT-max and T-opt from just the CT-min and B values, because T-opt is just 2/3 of the breadth and CT-max is breadth plus CT-min. So we were able to incorporate this into the forward evolutionary framework of SLIM. And from there, I also incorporated empirical time series data. And so we wanted to investigate if we were able to model seasonal adaptive tracking in using our model without invoking any specific parameterization such as seasonally switching dominance. So we compared it to empirical data that our collaborators at the University of Kentucky collected. And so first we incorporated the time series from a collection site in which they collected the wild Drosophila suzuki over spring and fall seasons. And so this is a temperature time series for that site. And this is the empirical CT-min that our collaborators measured. So they've had 3 sampling time points over a series of 4 years. And we're able to see that we do see CT-min tracking aligning with the temperature distribution. But however, we do see a slight lag with the CT-min trait following the temperature. And in summer, this is going to be a shorter lag, and the winter will be longer. And then we parameterized our model using their data of the CT-min extreme and CT-max extreme and other factors. And we were able to find that our model was able to capture the same CT-min trends, and we were able to capture both in 3 out of the 4 years, the similar CT-min values, as well as the lag as well. And in addition to capturing the values, we were also able to capture similar spread of data in our simulations compared to the empirical data. And you can see that the empirical has some more extreme values on either both ends of the CT-min, but this is possibly due to just phenotypic plasticity that our model was not able to incorporate yet. And so this is more of a deeper look into the lag associated with the different values of the thermal performance curve, such as like T-opt, B, CT-max, CT-min, going up the figure. And as you can see, the lag in the summer is shorter than the lag in the winter. And this is due to the longer generation times in the winter. But we are able to see that this lag exists because of the cumulative effect of the small effect loci affecting the different traits. And so other factors that are potentially affecting the seasonal adaptive tracking are generation length, autocorrelation, and genetic drift. So we were able to investigate this by looking at typically empirically, we can see that the Suzuki experience longer generation times in the winter due to entering diapause. And then in the summer, when they're experiencing the boom part of the boom bust phenomenon, they have shorter generation lengths, and the shorter generation lengths lead to their allowing for more generation times, generations over the summer, which leads to more rapid adaptation. And we also looked at the influence of autocorrelation on the ability for them to the evolution of the thermal performance curves. And in data with higher positive autocorrelation, we were able to see that they were able to track faster and more accurately. And then another factor is genetic drift, in which higher genetic drift makes it harder for them to seasonally adaptively track. And future directions that we could take this research is incorporating more realistic demographic patterns into the model, such as move and bust, population dynamics, and gene flow and migration across climate zones. We could also extend it to being a non-Wright-Fisher model, which would be able to incorporate effects like the temporal storage effect and buffering, which we know has effects on the ability for populations to evolve. And we could also extend to a spatially explicit model to forecast extinction risk and range shifts. And I want to thank Dr. Katie Lauderhouse and Jisun Min and the rest of the Lauderhouse Lab at Northeastern, as well as our collaborators, Joaquin Nunes and Nick Tietz at University of Kentucky, as well as SLIM and NSF for funding.
Thank you, Zoe, for your presentation.
Thank you.
If anyone has any questions, kindly use question and answer section.
Thank you.
Zoe, you can stop sharing your screen. As we have a few minutes, so we will start our session 3 minutes later. In the meantime, kindly, Darren Irwin, please upload your slide.
You can see that okay?
Yes, everything is okay. Kindly wait for 2 minutes and
Okay.
then we will. Thank you.
That was a very nice talk, Zoe.
Thank you. I'm excited to hear yours.
And Robert, you too.
Thank you.
Again, if anyone has any questions for Robert, they can ask right now, as we have 2 minutes more. Hello, you can present your presentation right now.
Okay, great. Thank you all for coming to my talk. I'm really excited to share with you some work I've been doing on an idea that I call the driving W hypothesis, and specifically how this relates to variation in mitochondrial DNA. So the study of variation in mitochondrial DNA has been central to the fields of phylogeography, taxonomy, and conservation genetics, among others. Despite its predominance in these fields, mitochondrial DNA is unusual compared to the nuclear genome because it's almost always inherited from only the female parent. And because it's inherited from one parent, and because there are many copies of it in a single cell, mitochondrial DNA can be used to construct genealogies in a really clear way. These are often used to infer history of populations using coalescent theory. However, this usually assumes selective neutrality, so the lack of any kind of selective process. So, yet mitochondrial DNA often shows surprising patterns. In one study from our own lab, we see that two well-marked species of bird share the same set of highly related mitochondrial DNA haplotypes. So here they share many of these haplotypes. And the combined variation is vastly lower than expected for different species. In contrast, nuclear DNA of these two groups is as divergent as expected for distinct species. This leads to the conclusion that there was rapid spread of one particular type of mitochondria throughout both species, facilitated by hybridization. Now, many other studies have seen similar patterns. It's quite a common thing now observed in the literature, and it's so common that there are review papers that compile these cases of mitonuclear discordance. A variety of explanations for low within and between species mitochondrial DNA variation have been proposed. These generally involve the lower effective population size due to uniparental inheritance, the lack of recombination affecting the entire mitochondrial DNA in the same way, and sex-biased dispersal. And in the last couple decades, there has been growing awareness of natural selection on mitochondrial DNA as a possible cause of low variation within populations. Another possibility is linkage to something under selection. In particular, in groups with a ZW sex chromosome system, such as birds, snakes, and butterflies, The W chromosome is inherited matrilineally in the same way as the mitochondria. So here we have females with ZW sex chromosomes and males with ZZ. The ZW daughters inherit their W from their mothers. The males have mitochondria that they get from their mothers, but they don't pass the mitochondria on. This means that the W chromosome here and the mitochondria are inherited in a perfectly matrilineally linked way. Realizing this, in 2007, Berlin et al. Made a fascinating observation. They observed that within species, mitochondrial diversity is about 3 times lower in birds that are ZW sex chromosome systems than in mammals that have XY sex chromosomes. They propose that this is due to selective interference among loci on the W, causing reduced effective population size. However, there are not many functional genes on the W, maybe calling this idea into question. Lane, 2008, proposed a different explanation, that flight physiology of birds causes intense selection on mitochondrial physiology. I'm now proposing that there's another process in which selection may be intense on the W regardless of what functional genes are present. And that process is meiotic drive. This is defined as an advantage that alleles can have in being transmitted from heterozygous individuals into their gametes. There are many known examples that have been well studied of meiotic drive, but they are mostly in autosomes or XY sex chromosomes. So what about in ZW sex chromosome systems? Well, a fascinating thing about female meiosis is that a single premeiotic cell produces only 1 egg after undergoing 2 cell divisions. So in meiosis 1, the duplicated chromosomes line up in homologous pairs, and then the spindle fibers pull one of each pair in each direction. One of the resulting cells becomes a polar body, which is a reproductive dead end for the chromosomes there. The other cell undergoes meiosis 2, where again one polar body is produced. Only the chromatids that end up in the egg get passed on into the next generation. So there's intense competition among these chromosomes up here to become these chromosomes down here in the egg. And it turns out that there's a molecular directionality to these spindle fibers, almost like a signpost saying this way to the egg for chromosomes that can read that signpost. Centromere variants that interact more strongly with the spindle fibers going to the egg can gain a transmission advantage. So what happens if a normal W chromosome mutates into a W with a centromere drive advantage? Well, then the driving W, indicated with the red W star here, then has a higher probability of getting into the egg because it can recognize and interact with these spindle fibers going toward the egg. Females with this driving W then tend to produce more W gametes than Z gametes, such that their offspring sex ratio is female biased. In this diagram, the driving W has a 50% transmission advantage over the Z, so it produces more female offspring with that W star. And we can see that between the first generation and the second, the W star is increased in frequency compared to the non-driving W over here. If we carry this forward to a third generation, we see the driving W has increased in frequency even more. And the key idea here is that whatever mitochondrial variant happened to be in the same female that had the driving W mutation will then spread along with the driving W, increase in frequency, spreading through the population. So if we now think at a population level, we have a whole species range on a landscape represented by this black shape. And the circles of different colors represent variants of mitochondrial DNA in the species. A driving W arises in an individual with the red mitochondrial variant, and that driving W spreads through the population carrying the red mitochondria with it. This dramatically reduces the mitochondrial diversity in the species. Now, if another species hybridizes with the first, the driving W can spread through the other species, carrying the red mitochondria with it. So I've designed computer simulations to evaluate the potential of W drive to cause sweeps of mitochondrial DNA. In these simulations, we have a large population of sexual diploid individuals, with each individual having a pair of sex chromosomes and a pair of autosomes. Each chromosome has a parameter M, which is the meiotic strength of attraction to the spindle fibers leading to the egg. Normal non-driving chromosomes have M equals 1, whereas driving chromosomes have M greater than 1. And the probability of a chromosome being passed from mother to offspring is the M for that chromosome divided by the sum of the M's of the two chromosomes in the pair. I use this simulation framework to answer 4 questions. First, what is the relative potential of selective sweeps for equivalent strength WZ and autosomal drivers? Second, do evolutionarily important W drivers necessarily have a large impact on the sex ratio? Third, does a Z driver reduce the potential of a W driver to spread? And fourth, how much potential is there for novel autosomal suppressors to prevent spread of W drivers? We'll see the results for each of these in turn. So first, it turns out that W drivers spread through the population much faster than equivalent strength Z and autosome drivers. So these figures show the spread of each type of driver in 3 different simulations where the driving version of the chromosome starts at a frequency of 5%, and the driver has a 50% greater strength of attraction to the egg compared to the non-driver. The driving W spreads faster than the others because the W always finds itself in female meiosis. It doesn't occur in males. Whereas the driving autosome here in green finds itself in male meiosis half the time, and the driving Z finds itself in male meiosis 2/3 of the time. And there's no advantage for the drivers in male meiosis where 4 sperm are produced from a single premeiotic cell. So there's no competition to get into a single egg like there is in females. Sometimes it's said that driving sex chromosomes might not spread much because they tend to cause a skew in the sex ratio, and that skew might be thought to select against the driver. But in the case of a W driver, they can spread while having only a subtle effect on the sex ratio. So here's an example with a driver with a 10% advantage that goes to fixation in an evolutionarily fast amount of time, while shifting the sex ratio so subtly here. This purple line is the females, proportion females. It's such a subtle shift that it would often not be detected in many studies of wild populations. Now, it might seem initially like the spread of a W driver would be countered by the evolution of a Z driver as an evolutionary response. This, however, is not the case. The red lines in these graphs show the spread of a W driver, which occurs a bit faster here at the top. It occurs a bit faster when there's also a Z driver. So these graphs with blue are a Z driver present, and the steepness of these red lines is a little higher when a Z driver is present. This is because the presence of a Z driver biases the sex ratio towards males, meaning there is even more positive selection on a W driver that produces more females. So an arms race between W and Z and their driving ability would simply lead to even more potential for W drive to cause sweeps in mitochondrial DNA. Finally, can the evolution of autosomal suppressors stop the spread of a W driver? To answer this, I introduced an autosomal gene that causes suppression of W drive. Suppression strength is S, and the total suppression is the average S from both autosomes. I'll note that in this model, there is no intrinsic cost or disadvantage to the W driver. Hence, selection for suppressors arises only from the biased population sex ratio caused by the spread of the driving W. In these simulations, we have a driving W and an autosomal suppressor, both starting with a frequency of 5%. The driving W, shown with the red line, spreads vastly faster than the suppressor, the gray line. This is for 2 reasons. First, when the driver and suppressor are rare, the suppressor and driver are only rarely in the same individual, so selection for the suppressor is quite weak. Second, selection for suppression is weak when the population sex ratio is near 50-50, which it is at the start of the simulation. One consequence of this second reason is that a complete suppressor, here at the bottom, may never go to fixation. See, this gray line sort of gets high, but it doesn't quite go to fixation, whereas a partial suppressor at the top does. And this is again because selection for the suppressor becomes weaker and weaker the closer the population sex ratio, here in purple, approaches 50-50. So we see from simulations that W drivers have great potential to spread. How much empirical evidence is there? Well, in zebra finches and chicken, W diversity is vastly lower than autosomal diversity, and birds have 3 times lower mitochondrial diversity than mammals. These observations are consistent with W drive causing matrilineal sweeps. Also, there's much evidence that birds have individual variation in offspring sex ratio and can even adjust their sex ratio based on environmental conditions. This suggests there are mechanisms by which the W or Z can preferentially enter the egg. W drivers could use related mechanisms. I think hybrid zones may be a perfect area to look for biases in soft offspring sex ratio because the drivers can escape the suppressors in those hybrid zones. And simulations show W drivers may be hard to detect because they can spread fast and the sex ratio distortion can be subtle, so we need to keep that in mind. So in conclusion, repeated cycles of W drivers are almost inevitable from a theoretical standpoint, causing low diversity of mitochondria. But there are a couple caveats. Direct selection on mitochondria and W variants might counter W drive, and W drivers might cause problems in terms of whole organism fitness. To learn more, please see my bioarchive manuscript here or this GitHub site. And I'm happy to take questions. Thanks for your attention.
Thank you for your presentations. If anyone has questions, you can use question and answer section.
I see a hand up. Ulrich has a hand up.
Yes. So my question would be, there are certain insect groups that have a extremely high female ratio, but do sometimes still have some male individuals. Do you think this could be connected to these W drivers?
I think it could if there's ZW sex chromosome systems. Also, in some insects, you have Wolbachia, which are female biasing parasites that have a similar effect as this driving W. So that's a good question.
Thank
Yes.
you for your question. You can use question and answer section for your question as we have limited time. So you may close your presentation, please. And our next presenter is Sarvesh Baheti.
Hi. Right, can you see my screen now?
Yes, definitely.
Okay, thank you. I'm going to present my undergraduate research that I completed in India, and this is a paper on fluidity and predictability of epistasis on an intragenic fitness landscape. It's currently a reviewed preprint in eLife, and because of the current model that eLife follows, it will soon be published there as well. I wanted to briefly introduce this topic. Let's say that you have a sequence with 2 alleles, A and B, and a mutation on both of these alleles can create a variant big A and big B. On a fitness landscape, you would have 2 different pathways by which this small a small v can lead to big A big B. And the only difference between these pathways is the order in which these mutations occur. So if we have to estimate the net effect of these 2 mutations, we are using Malthusian fitness here. So the net effect of those 2 mutations will be the selection coefficient of the first mutation plus the selection coefficient of second mutation in the genetic background where A the first mutation has already taken place. But because these pathways only differ in the order in which these mutations occur, if there is no epistasis between these two mutations, in that case, it doesn't matter whether the mutation B takes place before mutation A or after mutation A. So the null expectation for no epistasis becomes that the net effect of these mutations is simply the sum of the additive effects of individual mutations. And this is the idea that we used in our research to classify epistasis. So the rubric that we followed to classify epistasis was simply if there is no epistasis, this was our null hypothesis, that the net effect of the two mutations will be the null expectation, which is the sum of the fitness effects of these individual mutations. But this is not always true. And in some cases, if this null hypothesis was rejected, we checked if the net effect of these mutations was of a different sign than the null expectation. So if the null expectation was beneficial, we checked if the net effect was deleterious and vice versa. This is a very broad definition for sign epistasis. So we further broke it down into 3 different cases. If the double mutant had a higher fitness than the genetic background, we can count how many paths are blocked to Darwinian evolution. And if only one path is blocked, we can classify that as single sign epistasis. If both paths are blocked, we can classify that as reciprocal sign epistasis. If the double mutant had a lower fitness than genetic background, we can classify that as other sign epistasis. Using this protocol, we are only left with the cases of magnitude epistasis, where if the net effect of the two mutations is higher than the null expectation, we can classify that as positive epistasis, which also includes antagonistic epistasis. And similarly, if the net effect of these two mutations is lower than the null expectation, we can classify that as negative epistasis, which also includes synergistic epistasis. Now, the idea is just that given any two mutations, we can classify those two mutations as having one of these 6 cases of epistasis. Now for this study, we used the empirical data of a Foley fitness landscape. This is the fitness landscape that was constructed in 2023 by Andrei Popko and co-workers from the Wagner lab. This is a very extensive fitness landscape, and they created a fitness map of nearly all 9 base pair not all 9 base pair variants in a specific region of the Foley gene in E. coli. And we used this for our study. So the first step was to quantify epistasis in this entire landscape using the rubric that I showed before. And the idea was that simply if we start with a sequence, we introduce any 2 mutations in this sequence, and we try to estimate what's the nature of epistasis here. We can repeat this entire process for all backgrounds on which these 2 mutations can take place to get a fraction for all the different cases of epistasis observed across these different backgrounds for these 2 mutations. And we repeated this entire process for all the different pairs of mutations A and B to get a distribution of these fractions for all 6 cases of epistasis. We found that we repeated this process for functional and non-functional backgrounds separately, and we found that in most cases, in most backgrounds, we observed no epistasis. But in functional backgrounds, there was a significant proportion of positive and negative epistasis, which was not true for non-functional backgrounds. Now, once we had this in hand, we also wanted to study higher order epistasis. And more specifically, we were interested in seeing if a mutation takes place at a given locus, does that change the nature of epistasis between 2 mutations at totally different loci? And to check that, again, we started with a sequence, but this time we introduced a mutation at the locus that we wanted to study. And then the 2 mutations A and B, were introduced at different loci. We compared the nature of epistasis observed in both of these focal backgrounds. And we checked if the nature of epistasis changed because of the mutation at this locus I that we are studying. We again repeated this for all possible backgrounds and all possible mutation pairs. And we got the distribution of the fraction of cases where the nature of epistasis changed. So basically the probability distribution for epistasis changing here. And we found that again, there was a big difference between functional and non-functional backgrounds. But most importantly, in functional backgrounds, the probability of epistasis changing because of a mutation at any site was fairly high. And something that I would like to point out here is that at these locus 4 and 5, these loci were reported to be functionally important in the Andrei Popko's paper. And we found that in any mutation at these sites is even more likely to change the nature of epistasis. And this just showed that higher order epistasis is prevalent in functional backgrounds. Now, because we had a data set in our hand, we also wanted to study if the mutation that we are introducing was explicitly synonymous. Does that still change the nature of epistasis here? And we were really interested in this because we already know that epistasis can be highly idiosyncratic, but we did not know if synonymous mutations can also play a role in shaping epistasis at higher order interactions. So luckily, because we were using a 9 base pair sequence, introducing any 2 mutations leaves at least one codon intact. And our plan was to introduce a synonymous mutation at this unaffected codon and see if that synonymous mutation changes the nature of epistasis between the 2 mutations. Something that I want to highlight here is that we also consider changes in stop codon variants as synonymous change. So any change that was allowed at the second position in each codon was only a stop codon change because there is no other synonymous mutation available there. We also allowed that mutation in this case. So we repeated the same process as before, except this time we introduced a synonymous mutation at the site that we wanted to study. We made sure that the codon on which the synonymous mutation is taking place remains unaffected. The 2 mutations were introduced on both focal backgrounds, and we compared the nature of epistasis. We repeated this entire thing for all possible backgrounds, all possible mutation pairs, while keeping the locus of the synonymous mutation the same. And we got the probability distribution again. Surprisingly, we found that even synonymous mutations change the nature of epistasis at higher order interactions. And what was even more surprising is that if you look at these loci 2, 5, and 8, these are the second positions in each codons. The only substitution allowed at these sites was a change from one stop codon to another. So even changes from one stop codon to another change the nature of epistasis at almost the same rate in functional backgrounds, which was really surprising to us. And I think this was one of the most important findings in our paper, because previous work had shown that deleterious mutations can interact in unpredictable ways, but we found that even synonymous changes and stop codon and changes in stop codon variants can alter the architecture of epistasis. Synonymous substitutions, we know that synonymous substitutions can alter codon usage or mRNA folding or translation rates, which might be shaping the adaptation of other mutations somehow. But we theorize that maybe even premature stop codons, while they only truncate the protein, they might still influence some transcriptional or translation dynamics such that it reshapes the adaptive potential of other mutations by changing the nature of epistasis between those 2 mutations. And I think this is the only result I'll have time to discuss in this presentation, but I would like to thank my undergraduate lab members and Joanna, who's my current PhD supervisor at University of Arizona. And if there are any questions, I would love to take them. Thank you.
Thank you, Sarvesh, for your wonderful talk. If anyone has questions, you may ask. Thank you, Sarvesh. Our next presenter is Fahimeh Rahimi. You may upload your slides.
Can you see my screen?
Yes,
Okay.
you can start now.
Okay. Hi, everyone. My name is Fahimeh Rahimi. I'm a PhD student in John and Joe's lab at the University of Florida. And today I want to talk about a framework for detecting and characterizing epistasis across biological scales from individual proteins all the way to the whole genomes. I want to thank Sarvesh for the great presentation. I'm also going to give you a brief introduction on epistasis. So epistasis is anything like any deviation from an additive expectation. So for example, here you can see this gray dot is an additive expectation of these two mutations, and any deviation from that would be considered epistasis. And it's especially important in proteins because there are like 3D structures in proteins and residues are in contact in proteins. 35 to 70% of mutations in proteins show epistatic interactions. Also, you can see epistatic in polygenic and complex traits. For example, this is a classic example here. And 80% of missing heritability in complex diseases could be explained by epistasis. And it's also pretty important in agriculture. For example, 10 to 30% of crop heterosis is driven by epistasis. And these two papers here are showing that pretty clearly in yeast and wheat. So we know that epistasis is important and we want to measure that. A standard way to measure epistasis in proteins is DMS or deep mutational scanning. And the way that it works is that you can mutate to like one pair of residues simultaneously, and then you would measure their epistatic effect, and you could compare that to where you would measure their effect individually, and that would give you the epistatic effect. And here in this paper that they did the same thing, and they could measure the GB1 protein epistatic interactions, all pairwise interactions. Since it was a pretty small protein, like 56 amino acid, they could do this exhaustive measurement, and it was feasible. But imagine you want to work with something bigger. Once you want to work with something bigger, you will have more number of interactions to measure, and you will hit this red dashed line, which is a data ceiling. And so, for example, here, if you want to measure number of interactions in an average protein, it would be 10 million. And a large-scale QTL studies, it would be a billion. And in human GWAS, it could be 10 to the 13. So it would be quite not feasible and hard to manage. And that was only pairwise interaction. So if you want to look at higher order interactions, three-way, four-way, and higher order interactions, it would be exponentially worse. So this would create three challenges that you can see here. The first one is experimental limit that we talked about. The second one is computational cost. If you want to compute the pairwise interactions of like a relatively large data set, it would take up 1 year of CPU time. So that would be pretty costly computationally. But imagine you can get away with these two challenges and you can somehow fit the model. You will end up with the third challenge, which is the interpretability. So you will end up with a hair bar like this. So for example, this is a real genomic network in yeast. And so looking at this, how would you extract biological insights? Okay, so here's a study of this lab. I think this is the Michael Desai lab. And so they ended up with this kind of hairball, and if you stare enough in this hairball, you would see some genes that are interacting with a lot of other genes, and they call these genes epistatic hubs. So you could see some high-level structures emerging in these kind of like networks, but we need a very good pipeline so we can actually identify this kind of structures, and that is our goal. So in order to address that, we take a step back and instead of looking at every specific interaction, we ask another question, and that is how much does each of these individual sites would participate in epistasis, average over all genetic backgrounds. So that would give us this marginal epistasis for each site. So that would help us with the scalability issue because instead of L choose 2, for example, for pairwise interactions, you will deal with L marginal effects only. So only one per site. And that way, you can get higher order interactions at no extra cost. So you can look at pairwise, 3-way, 4-way marginals with the same L parameters. So this is the overview of our framework. So our framework is mostly in like interpretability framework, and so the way we apply this is that we take sequence function data and we apply a Gaussian process regression on top of that, which would extract pairwise and higher order interactions, and that would generate this full posterior. So you can predict the fitness for any genotype, even for the ones you haven't measured. And then we apply a family of kernel projections that would project this full posterior onto orthogonal subspaces that would give you different kind of marginals, like site marginals, type like different order of interactions, gene, pathway marginals. And so, as I mentioned, we were working with the full like sequence function relationship. And one thing to note that is that the genotype space is quite large, and there are like the possible number of genotypes are more than the atoms in the universe. And like we're unable to explicitly write everything down. So here's a result from machine learning called kernel trick, which would let us to work with the whole landscape, but it would scale only with a number of data points. So that would also help us with a scalability issue. And so before going and talk about real data, we did some simulation. So we simulated a landscape with known interaction structure. So we knew where the epistatic hubs are and everything like that. And then we applied our method and we compared recovered versus true marginal effects. So we applied our method, which is the Gaussian process, and also Planck, which is a naive, like exhaustive search. And as you can see, like our AURC are consistently higher than the Planck method. And another application was to proteins. For example, this is AAV2 capsid, DMS data. And we applied our method, and here's the result that you can see. These are the side marginal effects at each of these residues. And you can see, for example, these epistatic hotspot. And also for example, this site, you can see that there is a relatively weak additive effect, but you can capture this epistatic effect using this model. And this is just a 3-way 3D structure of that. And also using our method, you can actually zoom in and looking at these epistatic interaction, this epistatic hub, and you can see they have actually this 3-way interaction. So by, for example, mutating any of these residues, you would destabilize the other interactions. And here again, you can see that the variance explained by each order of interaction is pretty consistent among these interactions. And we applied the same framework to another protein, which is the GFP protein, the 4 orthologs of GFP. And this is only showing the pairwise interactions. And you can see this epistatic hub is shared within those. But for example, this one and this one, these are unique to one of the orthologs. And so this is the 3D structure of GFP, and there are like 11 beta sheets. So we also looked into the pairwise epistatis between those beta sheets. And we looked at the correlation between the pairwise epistatis and their distal position as 3D structure. And you can see that they're highly correlated. The closer the beta sheets, the higher would be the epistatic effect. And we finally looked at a comprehensive analysis of a large-scale yeast epistasis QTIL study, again from Michael Desai lab. And so we used their data set, which is 100,000 haploid segregants. They measured the fitness on 41 on these segregants and 41,000 SNPs across different environments. And here the phenotype is growth under heat stress. And so when I plot our model and when we added epistatic terms to our model, we saw that the R squared was moved from 0.6 to 0.69. So our GP regression model can actually capture epistatic effect, like a great deal of that. So we next want to apply our marginal epistasis framework to interpret the posterior distribution fitted by the model to understand the genetic architecture of this trait. So the first thing we looked at was the Manhattan plot. You can see here the QTIL. So the x-axis are showing the genomic positions, and the y-axis is the additive variance explained by these genomic positions. You can see the additive, pairwise, and third-order epistasis. So here you can see the epistatic hubs. And another thing to mention here is that, for example, for this case, you see that the additive signal is pretty weak, but you can see it is pretty prominent in pairwise and third-order epistasis. And because we are using this Gaussian process, we get a full posterior, and because of that, we can have uncertainty estimates for site marginals. And here you can see the uncertainty estimates for these epistatic hubs using the posterior samples and map solution. And because we're having this epistatic hub, another step we can take is to look at the interactions between these genes and even with chromosomes. So because looking at a chromosome would be less costly, we can first look at the chromosomes, but you can see overall these two are pretty overlapped. So this is, for example, interaction between chromosome 13 and 14. And you can see these two genes are in these two chromosomes are highly interactive with each other. And just like any QTIL study, we again have the linkage problem. And a way to address this linkage problem could be by doing pathway enrichment for epistatic hubs and interactions. And the key idea behind that is that it is unlikely for all the linked genes to the causal gene to exist in all the pathways that those genes are enriched for. So we did the pathway enrichment, and we found these top 10 pathways that are linked to stress and heat response. And when we compare that to the only main effect pathways with no epistatic pathways, we see that there is a little overlap between those, showing that how important it is to take into account epistatic interactions. And finally, we looked at pathway crosstalk between those epistatic, highly epistatic pathways. And for example, here's an example. You see that L-tyrosine degradation is highly interacted with this ubiquinone biosynthesis. And when we looked into the reason why, it's because it provides a precursor for this pathway. And with this, I want to thank my lab and my collaborators, and also the funding resource for this research. Thank you.
Thank you, Fahimeh Rahimi, for your wonderful presentation. If anyone has question, they can ask. Our next presenter is Tomar Zilbershtein. You may upload your slide.
Um, Hello. Just want to make sure the the correct slide is being shown.
Yes, definitely. You may start now.
Awesome, thank you. Um, Sorry, just setting my timer. Hello, thank you. My name is Tomer Zilberstein, and I am a medical student at the University of Arizona College of Medicine in Phoenix, with a passion for space medicine. Um, I'm here to discuss some of the research that I'm working on. I want to ask a question that sits at the intersection of evolutionary biology and one of the most consequential decisions that I think humanity will have to make this century. If we are to colonize Mars, um, will we become a different species? Not, of course, in the science fiction sense, but in the biological sense. Will our hybrid offspring between the Terran and Martian humans, be less viable, less fit? And so that's the question that we're exploring today. Um, so Mars gives us the complete checklist for allopatric speciation. We have a small founding population, strong directional selection on multiple traits simultaneously, and variable gene flow that is ultimately a political decision rather than a biological one, but with very real biological implications. Every one of these conditions exists in nature, but rarely all three at this magnitude in a single human scenario. The question is not so much whether these conditions are sufficient for speciation in principle, because we know they are. The question is where that boundary lies, given the actual parameter estimates that we have from human genetics. So the simulation is a forward time Wright Fisher model and run in Slim 5 over 30,000 generations, or roughly 750,000 years, sweeping across founding population size, migration rate, and mutation rate. Each cell getting 10 replicates, and we use Cox proportional hazards regression to identify which parameters dominate. So we have 6 chromosomes, 4 carry adaptive traits, 1 is a neutral autosome, and the X sorry. And the X is what generates Haldane's rule via hemizygosity. And so there are 5 parameters of interest in the model that drive it. We have the founding colony size, which sets how strong drift is. We have a multiplier on the human per base mutation rate, which sets how fast new variation enters. The migration rate, which is the fraction of Mars that's replaced by Earth migrants each generation, and I'll touch later on how that is one of the limitations of the model. P is a per pair incompatibility probability and the chance that any two divergent fixations form a harmful pair in a hybrid, and C is the fitness cost each incompatible pair imposes. And the key uncertainty is in P, which carries an order of magnitude in empirical range, and everything else is better constrained. So we have 8 scenarios, each isolating one mechanism. The key for the results are growth and pulse migration, as well as extended sensitivity analysis, where we looked at P holding everything else constant. So the Neanderthal scenario is mostly there as an internal consistency check to make sure that the outputs are sensible, given that there's not much, the priors are not really well defined. So we have 4 traits on 4 separate chromosomes. Each selection coefficient is anchored to a documented human sweep, but this is, our best estimate of what we can find analogs for. So EPAS1 for cardiovascular, and sickle cell for radiation, high latitude depigmentation for vitamin D, and the circadian clock gene variation for the day length mismatch between Earth and Mars. The separate chromosomes, matter. There's no hitchhiking between the sweeps. Um, and Speciation. So we use the OR snowball models. Speciation is called when the male hybrid fitness drops below 0.5. That threshold is chosen so that it's based off of the Coin-Orr post-zygotic isolation index, which is empirically validated across vertebrate hybrid systems. The key quantity here is the count of divergent near fixation. So as n grows linearly, each pair, right, it grows quadratically. So hybrid fitness collapses faster and faster as divergence accumulates. So at the calibrated PNC, you need approximately 165 divergent fixations based on that 0.5 threshold. Critically, males reach it first because of X-linked incompatibilities being fully exposed to hemizygous males but partially masked in heterozygous females. So just a quick note, these are preliminary results. We only went over about 1,000 cells of the 10,000 cell full factorial sweep. For the most part, the findings should be directionally consistent with what we expect to find in the full sweep, outside of a few assumptions that I will explain after we discuss the results. So a calibrated parameter speciation is rare. The one exception being the growth bottleneck driven by early strong drift. And the orange bar, that is the sensitivity analysis, which I'll discuss more on the next slide. The headline here is that the boundary is above us at current best estimates, but not far above us. So Palmer and Feldman estimated P at about 1,000th, but the empirical uncertainty spans at least an order of magnitude. The figure shows what happens as P increases above calibration. There is some critical P value above which speciation rate rises precipitously quite fast. Inside the speciation window at 10 times P, we already have 47% of runs speciating. And at 50 times the calibration value, we have 87% of runs speciating. Now, the median is at about 125,000 years. And I don't believe that these are extreme parameters. They are within the plausible empirical range for P. Additionally, the F1 criterion that we use might actually be conservative because F2 hybrids expose incompatibilities that F1 heterozygotes mask. So that is an assumption that we're making, but it should be a conservative one. Additionally, gene flow is the dominant predictor of speciation here. Larger in effect in the larger the population size, the larger the effect, mutation rate or selection strength. So one migrant per generation contributing genetically is sufficient to prevent speciation in the drift-dominated regime, with the caveat that, of course, we were testing relatively small carrying capacities for Mars, and those are fixed throughout the entire simulation, which is a bit unrealistic. That being said, the pulse migration result is the most striking. 0 speciation events across 180 runs, regardless of the duration. Suspected, I hypothesize this because early gene flow resets the clock at the most vulnerable window. Now, this model assumes replacement migration again, so that it's not additive, and the true threshold may require somewhat more gene flow than the estimate of 1. And as you could imagine, when you only when the migration rate is a large percentage of the carrying capacity, it's going to exert an over-leveraged effect. And so that could also be contributing to it. But as we do the larger full factorial sweep, this might still hold. It would be expected to hold, I should say. And so sex-linked incompatibilities disproportionately affect male hybrids. And 65% of runs where X-linked incompatibilities accumulated, male hybrid fitness fell below female hybrid fitness. This is significantly more than the 50% chance expectation, with a statistically significant to 1,000th. The mechanism is structural, of course. X-linked loci are going to be hemizygous males. Any incompatibility involving any X-linked allele operates at full strength with no masking. Female hybrids carry one Earth and one Mars X chromosome, which partially buffers the X-linked incompatibility. So this is Haldane's rule. It's a well-replicated pattern in speciation biology emerging naturally from the model, which another validation piece. So when speciation occurs, how long does it take? The median across all conditions is 17,600 generations and approximately 440,000 years at 35 years per generation, with the middle 50% of runs falling between 240,000 and 582,000. Now, I do want to point out that this chart is a little bit misleading because, of course, here the yellow bars are the extended sensitivity analysis, and that will bring the median down significantly when it's not our best estimate of it. Rather, you can see over here in the blue, red, and teal, these are the calibrated parameters. And as you notice, the modern human out of Africa line over here, so that shows we would not expect to see any speciation there, just as we do not see any speciation between humans that emerged out of Africa and ones that didn't 65,000 years ago. Whereas the Neanderthal split does line up pretty well with the distribution over here to the right based on the calibrated values. The growth scenario produces a counterintuitive result. We have a larger carrying capacity, producing more speciation, not less. At calibrated selection, no runs speciate regardless of colony size, but at twice the calibrated selection strength, the larger colony speciates twice as fast as the smaller colony. Again, these are very small colonies for this. This is a 50-person carrying capacity and 200-person carrying capacity. The reason is that I believe a larger population generates more mutations per generation and reaches incompatibilities faster once selection is strong enough to drive alleles to fixation. Now, of course, the model does not take into account at this scale, inbreeding could reasonably take an effect here, but it's not really it's not modeled, and so we are only isolating the effects of the speciation here. The early bottleneck period is the same in both. It is the trajectory, I believe, the points that differs. So, the colony size is a lever that cuts both ways. Larger colonies are more robust, but they're also facing higher speciation rates when exposed to elevated selection. And so now I just wanted to take a moment to discuss the various limitations and assumptions of the model. There are 5 key ones. Predominantly, the migration is replacement, not additive. So true gene flow threshold may require somewhat more than the estimate from before. F1 only criterion means that we're measuring the best case hybrid. F2 fitness depends on which allele segregate out. So, that depends on dominance. The X carry is only neutral loci, so fast X effects are not being captured. And P comes from Drosophila. It is the single largest uncertainty in the analysis, but it is the best estimate that we have. And everything else on this list matters for the full sweep for the full analysis and to make sure that things will agree not just directionally but qualitatively with the results that we have here. But those first 5 could be potentially push things directionally or quantitatively in the other direction, and will need to be resolved in the full factorial sweep. Just in closing, 3 things to take away. First, at calibrated parameters, the boundary is outside the simulation horizon and the model brackets correctly. We have 65,000-year-old population showing 0 isolation in the data and then 0 in the model. Neanderthal divergence at 600,000 years shows partial isolation in both. And secondly, the boundary is not far above us. If P is an order of magnitude higher than Palmer and Feldman estimated, which is within the empirical uncertainty, 47% of runs speciate within 375,000 years. And thirdly, the boundary is controllable. Gene flow appears to be the dominant lever, and this is very much a policy decision. Even with minimal biological exchange, we should be able to prevent speciation indefinitely, which is good news for us. But we expect the full complete parameter sweep to sharpen the boundary surface considerably and also to resolve those open questions that I discussed before. Thank you very much, and happy to hear any questions.
Thank you, Tomar, for your wonderful presentation. If anyone has any questions, they can use question and answer section.
Yeah, I agree. Again, that might be because of the relationship between or the over leverage of just one out of 50, 200, or even 2000. On a larger time scale horizon, if the carrying capacity grew significantly, something like in sci-fi where you have a whole terrestrialization of Mars, that I believe probably would not hold. But at least for that the early segment of exploration and colonization on Mars, I believe that this would still be relevant. Thank you, Brian.
Hello, everyone. As we come to the end of today's session, I would like to express my sincere gratitude to all the presenters and participants for their valuable contributions and active participation. I hope this conference has created a learning platform for discussion and future collaboration among researchers from different backgrounds and countries. Thank you once again for joining us and making this session successful. Wishing everyone continued success in their research platform. Have a wonderful day. Thank you.
Thank you.